here we're going to speak about A&P. A&P is the second hormone in question here as we're looking at regulating the reabsorption of sodium and potassium. Aldosterone was the first one, and that was secreted in the adrenal cortex. A&P is secreted by the heart, the atrial wall. So it is secreted in response to stretch on the atrial wall. And remember, the atria are our receiving chambers. These are the chambers that collect the blood volume that is coming back to the heart. So if it detects an additional stretch, which means that there's more volume coming back into the atrial because of that additional distension of the wall, it will secrete A&P in release. What this does is this increases our GFR by dilating the afferent arterioles and increasing flow going towards the kidney, which is going to increase filtration rate. It also constricts the afferent arteriole, which further increases the pressure of filtration that is moving across the filtration membrane. All right, if we increase the afferent arteriole and we constrict the afferent arteriole, we force more of that blood to be filtered across the filtration membrane, thereby increasing the GFR. All right. It decreases sodium reabsorption by closing the sodium channels on the apical membrane. And then it basically increases the amount of sodium that is excreted or lost or wasted into the urine. So it kind of acts in an antagonistic way as aldosterone. Whereas aldosterone is encouraging the absorption to reclaim that sodium back into the blood, A&P is down-regulating or decreasing the sodium channels, right, or closing those sodium channels and blocking the reabsorption, thereby increasing the excretion of that sodium into the urine. All right. So let's look at this. So whereas we talked about the trigger for aldosterone as being low plasma volume, dehydration, hemorrhaging, the trigger for A&P is the opposite. It's high plasma volume, high pressure, high stretch on that atrial wall. So the atria detects that plasma volume because of the increased stretch or distension on the atrial wall, and it results by secreting A&P. And A&P has several effects on the kidneys. It increases the afferent arteriole dilation and the efferent arteriole constriction, which together trigger more filtration force or increase in GFR. That is simply going to increase the amount of sodium that is flowing through the filtration membrane or being filtered, flowing through the filtrate, and eventually the amount of sodium that is excreted. Similarly, it basically directly blocks that sodium reabsorption by closing those sodium channels on the apical surface of the principal cells in the distal tubule and the collecting duct, and that stops that sodium from being reabsorbed, thereby also increasing its excretion. And then it blocks the renin release, so it's going to be, sort of going against the aldosterone response because it blocks renin release. It decreases the aldosterone production through all of the steps we described. It stops the activation of angiotensinogen into angiotensin 1, stops the conversion of angiotensin 1 into 2, and then angiotensin 2 from triggering the release of aldosterone. So several steps in between renin and aldosterone. Remember, we described this, the RAS system, the renin, angiotensin, aldosterone system. And so it basically blocks all those events and blocks aldosterone, which blocks sodium reabsorption. So not only does it have its own mechanism to close sodium channels and stop sodium reabsorption, but it blocks aldosterone's mechanism and also indirectly stops reabsorption and increases sodium excretion as well. Alrighty. Which our third objective here is to explain the role of aldosterone in potassium balance. So moving on to our other important electrolyte, potassium. And again, starting off with some important terms relevant to potassium state. So hyperkalemia, high plasma potassium, hypokalemia, low plasma potassium. And the role of potassium is similar to what we described for sodium, is important to our excitable cells. It is the basis for establishing the resting memory potential and thereby exciting the cell to bring about the action potential. And so, especially when we talk about that um, hyperpolarization phase, repolarization phase, potassium's movement is so important for that phase. Let's talk about how potassium is handled. So it's freely filtered across the glomerulus, reabsorbed in the proximal tubule, and then in the distal tubule and collecting duct, it is both reabsorbed and secreted. Unlike sodium, sodium is only reabsorbed, or we can block its reabsorption, which is what potentiates its excretion, but it is not secreted. Potassium, on the other hand, is both reabsorbed and secreted. So we can actually move potassium out of the plasma, out of the cell, and back into the tubule and allow it to be, um, eliminated in the filtrate. So potassium secretion happens in the distal tubule and collecting duct, regulated by the hormones we'll describe. So aldosterone, and then it is basically stimulating its release, which is, as we have more potassium in that filtrate, we're going to trigger more aldosterone release, which is going to facilitate more of that excretion or secretion of potassium. So here's our principal cell once again, the apical surface. And the mechanism for moving potassium into the cell is still not quite well understood, not very, not well known. But we know that potassium does get into the cell by some mechanism, and it has to do so actively against its concentration gradient. So it moves into the cell actively, and then it can leak out of the cell passively, and then it can move across into the plasma. It is also being pumped into the cell actively on the basolateral surface by the sodium potassium pump, which explains why it needs to move into the apical surface against its gradient, because there's higher levels inside the cell. So now we think about its movement. It can freely move across the cell, but it can also move paracell or in between the cells as well. And it can leak in between these channels here and also make its way into the plasma. In the distal tubule, it's a little bit more straightforward. Here, we move it into the cell according to the sodium potassium pump on the basolateral surface, and then it can freely move according to that gradient, higher levels in the cell, lower levels in the filtrate or in the ECF, and it can freely passively move into that filtrate. So notice how it is moving in the opposite direction here, which is secretion, and that is going to be regulated by the levels of aldosterone. So our final checkpoint question here, which hormone regulates potassium secretion? What are some stimuli for the release of this hormone? So the regulation is basically by aldosterone. It can directly, if there's too much potassium in the plasma, in the filtrate, it can directly stimulate aldosterone release, and that can encourage its secretion, and it can be released into the urine. Okay. And just going back to some of its triggers here, its triggers are going to be an increase in plasma potassium, as well as an increase in the amount of potassium in the distal parts of the tubule, right? In the collecting duct and even the distal convoluted tubule. So those stimuli will directly trigger aldosterone release, which will encourage its secretion.